welcome to today's seminar, which will be covering the immunology of the inner ear. I'm Ellen Ryan. I'm a professor of otolaryngology and neurosciences at University of California, San Diego, and I'm also a research scientist at the San Diego VA. I study the mechanisms of hair cell damage and also immunology of the middle ear as it's related to otitis media. But it's a real honor and a pleasure for me to introduce my friend and longtime colleague, Dr. Elizabeth Keithley, who is our speaker today. So Betsy is an emeritus professor now at UCSD and the Department of Otolaryngology, and she joined the faculty here in 1985. She got her PhD in anatomy from BU in 1980, where she investigated age-related changes in the inner ear. She then went on to postdoctoral training at MIT and the Eden Peabody Lab, where she studied the physiological activity of neurons in the cochlea. And during her career at UCSD, she continued to study age-related hearing loss, but she also studied, and did many studies on the immune responses of the inner ear. She retired from UCSD in 2010, and she now serves as the chair of the board of directors of the Hearing Health Foundation, which is, a friend to many of us and launched, many of our careers with, new investigator grants. So anyway, before we get started, I wanted to talk to you a little bit about questions for this particular talk. If you have any questions for Dr. Keithley, please direct them through the chat to me, and then I can relay them on to Betsy after the talk is completed. So with that said, we're ready to get started. So Betsy, take it away.
Okay, can you hear me?
Yes, we can.
Okay, good. So I want to thank you all for attending this. I'm really pleased to be invited by the Education Committee, and I'm very grateful to them. And also to Don Keegler and Rhiannon McNeely, who put this together and helped me do all this technical stuff. So now we'll see if we can do this. So I'm going to share screen right now. Okay.
Yep.
All right. Thank you very much. So immune mechanisms of the inner ear. And why do we want to study this? To understand hearing in all its complexity. The immune responses have not been a very important part. It's only recently that we've recognized that there are immune responses and that they're important to hearing. And so we really know very little, but defining what they're doing and how they interact with how we hear will lead to better understanding of hearing loss and hopefully help us with helping people with hearing loss. So the immune function in the ear involves the inner ear, the immune cells, systemic circulation, the vasculature and lymphatics, lymph nodes and diffuse lymph nodes, the bone marrow, the thymus and spleen. So basically a lot of different parts of the organs in your body are all involved in this. And traditionally in classic immunity, they divided the activity of the immune system into the innate immunity, which happens in the early part when you're exposed to a pathogen. So it's between the first 12 hours of exposure. And macrophages phagocytose and in conjunction with complement get rid of cells that are infected with a virus or a pathogen, and natural killer cells are involved in that. But over time, after about 24 hours to 48 hours, you begin to develop adaptive immunity, which is the activation of B cells who now can make antibodies against the pathogen, and T cells, which kill and interact with the killer cells of the body. So now you're going to, and this is the long-lasting response that you have. But now, more recently, we've looked at tissue-resident macrophages, and these are multifunctional supervisory cells involved in homeostasis and tissue repair that monitor tissue environments to initiate and generate appropriate cell signaling cascades. These are pleomorphic, and the macrophages within tissues are pleomorphic and capable of changing shape and migrating in response to environmental signals. And these macrophages also have diverse gene expression patterns to generate cytokines and other molecules that help deal with pathogens. So they're a very complicated set of cells that are in your tissues. So where are they in the inner ear? In the cochlea, they're everywhere. They're in the stria, the spiral ligament, the scala tympani, and in the periosteum and the basilar membrane. In the medullus, they're the limbus, osteospiral lamina. They're in the round window membrane. And in the vestibular system, we know much less about them, but they're in the macula, saccule, and utricle. And then the endolymphatic duct and sac. So first of all, I want to thank Jennifer O'Malley, who provided these images for me. These are her sections from celoid sections of the human temporal bone, and they're stained with ionized calcium binding adapter protein, adapter molecule 1, so IBA1, and this is expressed in the tissue resident macrophages. So you can see in the stria vascularis that you have many cells labeled in here, and also in the otic capsule. You can see a cell labeled out here. And again, they're different shapes. You find cells that look like this, you find cells that are around this blood vessel, and then a more globular cell here. And this is another stria vascularis macrophage labeled here. And now this is another section from, this is Hainan Lang, who I also want to thank very much for providing this micrograph for me. This again is IBA1 positive cells. They're in the red. The green is the endothelial cells. So this is a stria vascularis that's been laid out. It's not a section, it's dissected out and laid flat on the slide. And it's this beautiful demonstration of the macrophages surrounding the endothelial cells, the capillaries and the stria vascularis. And then Dr. Shi at the Oregon Health State University has generated this schematic of the stria vascularis capillaries. And the endothelial cells are connected by tight junctions, and that's what forms the blood-brain barrier there. And like all epithelial cells, they're surrounded by a basement membrane. And then on the outside of that is the macrophages that, if anything, any macromolecules, pathogens, organisms manage to get out of the blood vessels, the capillaries, the macrophages are there to endocytose them and destroy them. So what you have is an incredibly complex system to keep the stria and the endolymph and the spiral ligament from getting infected with foreign anythings. So what about the spiral ligament? That's another highly vascularized and full of macrophage tissue. And you can see this is from a mouse with anti-F480 antibodies, which is activated macrophages labeled. And you have cells down here in the type 2 cell areas, the fibrocytes, type 1 fibrocytes. You can also see that again in this stria vascularis here. And this is a section from Jennifer O'Malley's human temporal bones. You can see again lots and lots of macrophage in the spiral ligament, and again pleomorphic, very different shapes in the different cells. And this is a section from a figure from a paper by Hu et al. And it's interesting to me. So this is looking at the basilar membrane from underneath. So you're looking up at the basilar membrane in scala tympani. And so that these cells are underneath it. And in the apical turn, you can see that they're very fibrillic. And as you get towards the basal turn, they're more globular. And, why is that? Why is that shape difference? I guess I would speculate that it has to do with the micromechanics of the organochordae, that it helps to have them in a different shape. So now comes a touchy subject here. Are there, in fact, macrophages in the organochordae or not? And in the human, this is a section again from Jennifer O'Malley's collection. This is a radial section, and over here you can see the pillar cells. This is the basal membrane down here. You can see there's some cells in the basal membrane, but there clearly are also in the epithelium some labeled cells. So it looks like there's macrophages in the human sensory epithelium. This is you can see the pillar cells here, inner pillar, outer pillar, and a huge macrophages in the organ, or in the tunnel of Corti. But you can also see the basal membrane macrophages down here. But if you look at non-human mammalian cochleas, you don't see macrophages in the organochordae in the sensory epithelium. And Antonen in 2014 and Lee et al. In 2021 published really beautiful papers showing that the hair cells, when they die from either acoustic trauma or ototoxic damage, are phagocytosed by the Diter cells, that the processes of the Diter cells up against the reticular lamina close in on the dying hair cell, and the hair cell goes beneath and is phagocytosed by the Diter cells, keeping the reticular lamina sealed at all times. And so it happens very quickly, and you wouldn't want first of all, a macrophage is too slow to get in there and deal with that. But second of all, you wouldn't want a macrophage in there messing around with this beautiful structure. So I think this is a really unresolved and deserving of more experiments to figure out what exactly is going on in here when this happens. In the medullus, you've got, again, lots and lots of ganglion cells, or lots and lots of macrophages among the ganglion cells, the nerve fibers, around the vasculature. And this is a higher mag among the ganglion cells. This is again a micrograph from Jennifer O'Malley, showing the macrophages among all of the neurons in the Rosenthal's canal. So the vestibular system is the poor cousin of the cochlea. There's much less known about it, but Kerr et al. Have been working on looking at the immune activity in the vestibular system, in the saccule and the semicircular canals. And among those epithelial cells, it's not quite as organized as the cochlea is, and there are macrophages among those cells, and they do seem to be phagocytizing the dying sensory cells. So the vestibular epithelia and the cochlear epithelia look to be working differently in that situation. Okay, so what about the endolymphatic sac and duct? This is not very often thought of anymore. It's people looked at it a long time ago, but you can see in this section here that the cells are stained for macrophage. There are macrophages in the lumen. And this is a diagram from Physiology of the Ear by Jan and Satosaki. And I like it because it shows that it's a very nice illustration of the endolymphatic space and the paralymphatic space in the inner ear and the semicircular canals. And this endolymphatic space, these cells continue on here and form a blind ending in the dura of the brain. So, what is this all about? First of all, I'm going to say that the lining cells are continuous. So in the lining cells of the duct and the sac are continuous with the lining cells of scala media, and those are epithelial cells, whereas the subepithelial tissue is the continuous with the perilymphatic space, and those are connective tissue cells, like the and so all of those cells continue out and form that sac. And the sac is next to bone marrow. And if you create an immune response, you can see some bone marrow cells moving into the sac. So the sac is sort of got a close connection with white blood cells and immune cells. The lumen contains the normal high sodium fluid, and I'm not going to get into what that's all about, but what it means is that cells can live inside that fluid as opposed to in the endolymphatic space in the cochlea, where it's a high potassium fluid and cells can't live in that environment. So there's macrophages, lymphocytes, plasma cells, and cellular debris if you look inside the lumen. And in the connective tissue, you've got the whole gamut of immune cells: monocytes, macrophage, lymphocytes, plasma cells, PMNs, and blood vessels, and most importantly, fenestrated vessels that were seen in the electron microscope, but they're likely to be lymphatic vessels. So with lymphatics, you can connect to the systemic immune system. So let's say a little bit about the endolymphatic sac in the cochlea. Antigen injected into the scala tympani reaches the sac and is phagocytosed there. Bacteria injected intrathecally reach the sac and the cochlear act, and they're phagocytized there. So bacteria getting in there are phagocytized, and antigen is also there. So what's the relationship between the cochlea and the endolymphatic sac? Experimentally induced inflammation in the cochlea causes ICAM expression on the endolymphatic sac vessels. So if you create an inflammation, in the inner ear, the endolymphatic sac knows about it very quickly, and ICAM expression on the vessels induces influx of inflammatory cells from the circulation. So expression of pro-inflammatory cytokines, IL-1 beta, IL-6, TNF alpha, and are in some of the inflammatory cells that are there. So there's already sort of a stirring up of trouble when you create something in the cochlea. IL-1 beta expression, this is a graph from Sato in 2003. IL-1 beta expression is induced by the cochlear immune response. So in the cochlea, you can see starts right away, obviously, and then goes out to 168. But you also get IL-1 beta expression in the endosac lumen and in the perisaccular connective tissue cells. So again, you're sending out signals quite rapidly from the endolymphatic sac. So with its vascular, lymphatic, and immune cells operates in a coordinated manner with the inner ear immune responses. So what about the vasculature? Because vasculature is very important to immune responses and lymphatics. So let me just take a minute and review. I'm sure you all have seen this figure and know this, but I'll just say it again. The spiromedullary artery enters the cochlea, branches come across between above the scala vestibuli and over to the spiral ligament, where they branch into capillary beds in the stria vascularis and coalesce. So now there are venules in the lower part of the spiral ligament and back to the posterior spiral vein to leave the cochlea. But the important part here is this intense capillary beds out among the stria vascularis and the spiral ligament. And that's the place where throughout the body where cells extravasate from the circulation into the tissues. And so you see it here. It's a scala tympani of a guinea pig with an experimental inflammation created there. And you can see the cells coming out here. And another point is that you can't float, or cells can't swim. So a matrix is very rapidly formed here. And while all that happens, it clogs up the blood vessels upstream from that. And so you get expanded of stasis and expansion of the capillaries up in the stria. And here's a higher mag of cells leaving the blood vessel and the scala tympani. Immune inflammatory cells interact with the extracellular matrix proteins and create that inflammatory matrix. Fluid-filled paralymph is a unique environment. I guess I already said that. But I think this is another area that should be examined, is how is this matrix formed? What cells do it? And we all know that you can get an ossified cochlea, especially with cochlear implants. You see that a lot. But how is this all formed and regulated? I think we don't know anything about that yet. So now let's look at lymphatic drainage. So when I started this business, there was, ears didn't have lymphatics. But if you put an antigen inside if you inject it into scala tympani, it's rapidly taken up by, in the spiral ligament here, the periosteal cells, and probably into this vein as well. And if you look 15 minutes later at the deep cervical lymph node, you can see labeled cells in the node. And this was just HRP that we put in here. 15 minutes later, it's also in the spleen. So whatever happens in the inner ear is very quickly recognized, or at least the fact that it's there. I don't know that you recognize what it is, but the systemic immune system is aware that something happened in the cochlea. So that's just saying the same thing quickly there. All right, so what happens in the other way around? What happens if you have a systemic infection? You have a cold, you have COVID. Does the ear know about that? So you can model that by putting lipopolysaccharide, which is an endotoxin. It's a cell wall component of gram-negative bacteria. You can inject it intraperitoneally and use it to simulate a systemic infection. And if you do this, it induces cytokine secretion, adhesion molecule expression on endothelial cells. And in the cochlea, vascular permeability is increased. Type 2 spiral ligament fibrocytes express NF-kappa B, which is an innate immune activator. Type 1 spiral ligament fibrocytes express IL-1 beta. CD45 leukocytes enter the scala tympani, and noise damage is increased. So think about going to a rock concert when you have a cold. It's probably not as good as if you don't have a cold. So this is just some illustration of that. 72 hours after injection of LPS, nothing else has happened. You just inject LPS intraperitoneally and you get labeled fibrocytes with cells that expressing IL-1 beta, which is a pro-inflammatory cytokine. So right away, things are happening in your inner ear. Inflammatory cells move into the inner ear. So you see some cells in here. Okay, and activation of these. And again, if you inject, so now if you inject antigen, so you put LPS intraperitoneally and then put antigen that the animal is sensitized to intrathecally, so you haven't touched the cochlea, you get increased number of leukocytes in the sections. In intrathecal injections, you get a few, and if you put LPS intraperitoneally, you get a lot. And you can see that over here. There's a few cells here that come in with the intrathecal injection of antigen, but there's a whopping number of cells here if you put antigen in there and LPS intraperitoneally. So I think this is a very unappreciated part of inner ear behavior, but if you have an infection systemically, the inner ear is aware of it and reacts to it. So I'm just saying this over again, is that activity in the cochlea is constantly monitored by systemic immunity, and conversely, systemic immune activity can induce by infection that affects the cochlea. Okay, so what are the immune responses to acoustic trauma? So that's the big stressor that we're all familiar with. And so what's going on there relative to the immune system? Macrophages assist in repair of damaged synapses between the hair cells and the spiral ganglion cells. So this is synaptopathy. We know with acoustic trauma that you get synaptopathy, but it does look like macrophages can move in from the osteospiral lamina and go in and help repair that. So that looks like a good function of macrophages. There's upregulation of pro-inflammatory mediator, NF-kappa B, and there's secretion of pro-inflammatory cytokines such as TNF alpha, IL-1 beta, IL-6, and chemokines, MCP1, MCP5, and CCL2, and recruitment of inflammatory cells, circulating inflammatory cells. So every time you have acoustic trauma, all this activity goes on. Okay. So innate immune activity is activated by acoustic trauma, and this shows this was a mouse cochlea, and NF-kappa B is activated in the type 1 fibrocytes and the type 2 fibrocytes, and this was a study done by Adams. Okay, and F480 is activated, macrophages, they're increased, and CD45 leukocytes come in. And this is a figure from Fry et al., 2018, and this is the same group that I showed you originally, the basilar membrane macrophages below the basilar membrane. Here you can see it with acoustic trauma that the number is increased in the area of the acoustic stimulus. But it's not clear exactly what these cells are doing during acoustic trauma, but they're certainly increasing their numbers. And this shows you inflammatory cell. This is a cat cochlea. It's a celoid and H&E stain section where that just seems really nicely illustrated that you can see these inflammatory cells moving in from the circulation. This is scala tympani. And this is, again, combining the antigen model where we inject the animal with an antigen and get him stimulated to that, and they have circulating titers against the antigen. So we put the antigen alone, and then if you put antigen plus noise, you get a much larger response to that antigen. So in conclusion, inner ear immune activity is an exciting topic of investigation. I hope I've given you a few ideas about things that certainly need to be investigated. And it's not isolated from the systemic immune activity, but affects it and is affected by it. And immune surveillance is a constant occurrence. And I'd like to thank, this is just some of the collaborators, but I mostly want to thank Jeff Harris and Alan Ryan and Gary Firestein, who have been my colleagues in these studies, and Hainan Lang, who provided me a beautiful micrograph, and Jennifer O'Malley, who provided so many of those micrographs for this talk. So thank you very much, everybody. And I'll take questions if.
Okay, thank you very much for that presentation, Betsy. It was wonderful. There have been a couple of questions that have come into the chat. So what I can do is relay those to you. Dr. Lafond asked about the different shapes of the macrophages. You know, the ones who are sort of fibrillary or elongated in the apex versus globular at the base. Could the ones in the base be more likely to be activated, an activated form, because they're closer to the round window membrane and more likely to be exposed to middle ear immunogenic materials or mediators?
Certainly could be. You know, a long time ago, and I couldn't find a picture of this. I probably had a slide, a 35 millimeter slide, but anyway, the round window membrane has immune cells in it, has macrophages in it, and certainly changes if you create a middle ear infection. The round window membrane gets thicker and cells move into there. So yes, it's very responsive, and yes, those cells could be from there, have migrated in, yes.
Okay, and then Anna Lisakowski also asked you that when you say that macrophages assist in the repair of damaged synapses in the cochlea, how do you think that works? Do they just trim them or remove them? You know, what is the form of repair?
You know, I think that Tajmir Kaur is going to give a talk in the future, and she probably will talk about that. I don't know. There's a publication that I've read, but I don't know how it works. But clearly that the macrophages are going in there and modifying something that helps the repair.
All right, and Royce Clifford asked, what would the inflammatory response be in Meniere's disease? Do you think that it's a cause of Meniere's or an effect of it? That's a tough question.
You know, I have very studiously avoided any kind of clinical interpretations with this. You know, I wish we had better anatomy, of better, you know, more pathology to look at from Meniere's. You look at a Meniere's temporal bone and it's been, you know, 30 years. So we don't have really good models of animal models of Meniere's. So it's very possible. I mean, my thinking is that it's very possible that it's an acute inflammatory response that gets cured, that, you know, that goes away, that gets resolved. And so that would surely give you a fluctuating, you know, it's certainly plausible that immune activity is happening in Meniere's disease, inappropriate immune activity that other people don't experience and the people with Meniere's do. It fits very nicely to that, but unfortunately we don't have good pathology from a human, so you can't, and you can't create a model that you're convinced of that that's how it works in an animal model. So that's where we're stuck.
Okay, then we have another question from Laurel Carney, in that you mentioned several times that macrophages move into an area of the inner ear. And it seems like they move around in the inner ear quite a bit. But what is their lifespan? And after they do work at one place, do they return to the circulation and the home tissue, or do they die and get replaced by new cells? Is anything known about their life cycle? And does it change with aging? So it's a multiple part question.
Oh, wow. Thanks, Laurel. So, no, I don't think we know a lot about the lifespan of them. They clearly move around. They're very mobile cells. And, but I don't know if they go back into the circulation and recirculate, or if they die in the tissues. I don't even know if it would be interesting to do a BRDU study to see if the ones in the cochlea divide, or whether when you see a greater number of them there, is it because they've come in from the circulation? I think those are all really wonderful questions, and I don't know the answers.
Well, based on our work in the inner ear, I can add a little something. Macrophages can live for a long time, sometimes for years. So the likelihood is that, unless they're damaged in some way by what they phagocytosed, they're going to leave the tissue, and go back into the circulation, and may end up somewhere else doing another job. They're sort of the handyman of the immune system, running around, doing things when they're needed. So anyway, let's go on to another question. First of all, Jennifer O'Malley wanted to thank you for, thanking her and for all of the images that she sent. And then Jarnail Singh asked if infiltration of systemic macrophages helps protect cochlear hair cells from noise-induced damage.
I don't know. I mean, that's the million-dollar question. We don't know the answer to that. So see, I told you, it's a very exciting field. We need the people to answer these questions, to do some more research.
Yeah, no question about that. We know that, M2 macrophages, which are homeostatic and anti-inflammatory, are, tend to be elongated. And a lot of the pictures that you showed were elongated. Whereas in the base, they had some rounded ones within the organochordae, which is, sort of the classic shape of the, M1 macrophage, which is pro-inflammatory. So do you think that, these elongated ones that we see, throughout the tissue of the inner ear are homeostatic ones, or is it just that they're forced to do that by, the torturous spaces they have to live in?
I mean, they look homeostatic because they are, if you just take a normal ear and perfuse it with no, no trauma, you see them. So I would say they're homeostatic.
Well, I think that's a reasonable conclusion. So you think that the ones in the base are not as homeostatic? That's why there's more damage in the base, because they're being bad actors.
That's certainly something that comes to mind, but I think it must have more to do with just the constraints of the mechanics of it. Ask Bill Brownell. You have to have you have to make everything that you're asking to work there to also be micromechanically workable. Mm-hmm. Whereas in most other tissue spaces, you don't have that constraint on you. You can be any shape you want. Mm-hmm. Doesn't matter in your skin or in your other organs. But in the ear, it matters.
That's a good point. So then finally, Jennifer O'Malley has a question. Do we know any more about where the lymphatics are located? In the human, she has seen ramified, elongated macrophages extending processes into the areas of the inner hair cell. She thinks they're homeostatic a lot of the time and then become activated when there's some kind of damage. But do we know where those lymphatics are located?
No, and we tried to use antibodies that labels lymphatics, but they're not very. I was never convinced that they weren't also labeling venules. So venules and lymphatics look like you can't differentiate them in an EM. You can say that there are cells that are fenestrated. You can see fenestrations, but you can't know whether that's because it was a venule or a lymphatic drainage vessel. So we need better labels for lymphatics, and I know that they're in the central nervous system. They're grappling with this as well.
All right, so Laurel has another question. It was mentioned that the macrophages return to the circulation. That suggests that they may act anywhere in the body, correctly? Are there any specialized immune cells that stay in the cochlea or are relatively general purpose, which could relate to the discussion about the special circumstances of the ear for mechanics and the delicacy?
We don't really know if cells are moving in. I mean, we know that cells move into the ear because you can see there's a greater number. But are they divided cells or did they move in? I don't think no one's really looked at that. I mean, you're going to have to do something like BRDU or label macrophages in some lymph node and then see if those macrophages move into the cochlea. So I think the answer is we don't know.
Okay. And then Jean-Francois Lafond has another question. When corticosteroids are used for acute treatment of noise-induced damage, there is certainly an effect on the macrophages. So their synapse repairing work could be affected. Would it be interesting to study that as well?
Yeah, it would be. That's a good idea.
Okay, and then Jennifer again says, when working with Bob Kimura, we identified macrophages near the sac. And she means the lymphatics. So she was able to see the lymphatics. I don't know how, but. Transmission electron microscopy, she says. So just a comment.
Okay. Yeah, I think there's definitely lymphatics in the connective tissue of the endolymphatic sac. And the original study that was published, or an original study, was Helge Rask Andersen and Wackham, Ashley Wackham. There's an EM study but they were careful to say that they were fenestrated vessels. They did not say that they were lymphatic vessels.
Okay, and then we now have two individuals, Dr. Lafond and Dr. Carney, who thank you for a wonderful presentation and a fascinating subject to talk.
Thank you.
And I would add that was just fantastic. So thank you very much.
Okay. Well, I just have to say, don't go to a rock concert with a cold.
Good advice for all of us who are rock concert fans. I'm sure there are many of us here who have them scheduled right now and may have a cold. So please be careful.
Okay. All right. Well, thank you all very much.
Thanks to everyone for showing up, and thanks especially to Betsy for this talk.
Okay.